This is Mr. Danko with the first of a series of review videos that I'll be publishing on Facebook for my students and the other students in physical science to use as a means of review for not only quizzes and tests, but for the end of course exam. The first is going to be on the scientific method. The scientific method is fundamental to science. It's how we go about finding new information and making sure that information is good. It's a several-step process. I'm going to go through 7 in all. The first of which is to identify a problem. During this step of the scientific method, we're simply looking for a question that we're trying to answer. There are some limits. Those questions must be based in fact, not opinion. This means we have to learn to think with words like most popular as opposed to best, or least popular as opposed to worse. The second step in the method is research. This doesn't mean little people running around in lab coats. This means going to the library, going to the internet to find some background information. We need to make sure our information is good information. To do that, we make sure our sources of information are accurate, credible, and relevant. Accurate's the easy one. Accurate means correct. We want to make sure our sources of information are correct in what they're telling us. Credible means believable. The opposite of the word credible is incredible. Incredible means unbelievable. For our sources to be credible, they need to be written by experts, people who know what they're talking about. Relevant. Relevant means related or current. We want to make sure that our sources of information are related to what we're trying to discover, what we're trying to investigate. We want to make sure they're current. We don't want to use sources of information that are too old. In science, things change quickly. We want to make sure that our sources of information are no more than, say, 5 years old. Third step is to form a hypothesis. Most students know what it is. It's an educated guess. It's the research step that came before that make us educated. We need to make sure that our hypothesis is valid. Valid does not mean correct. Valid simply means usable. To be valid, it has to be based on research or background knowledge or prior knowledge. If you already know enough about the question, you don't have to go to the library, you don't have to go to the internet, you can form a hypothesis. If you're lacking in that knowledge, that's where the research comes in. That hypothesis has to be testable. It has to be something we can actually collect data on. In science, it's all about the data. If you can't get it, you don't have anything. To give you an example of how this works, we can't say things like, if Promethean boards were in classrooms 30 years ago, adults today would be smarter. We can't go back in time and put Promethean boards in classrooms. That is not a testable hypothesis. The next step, step 4, design an experiment. When you design an experiment, you're going to be using variables, constants, and controls. There will be 2 variables in the experiment, independent and dependent. Independent variable is the one that you, as the researcher, can manipulate. You can change it. The dependent, you can't change. The dependent changes because of the independent variable. It responds to what you do. 2 terms often used to describe the independent variable and dependent variable are manipulated and responding. The independent variable is the manipulated one. The dependent variable is the responding one. Constants, sometimes called control variables. These are things that remain the same through your experiment or the same from one subject to the next. These include anything that can affect your outcome, that could change your results. And finally, controls. Controls are your standard for comparison. What you will compare your experimental results to. Your proof that a change took place. We'll take a quick look at an example to make sure that we understand this, to make sure that you can see how it's applied. Let's say I want to know if a new study guide that I made will improve test scores. I can design an experiment to test that. My independent variable in my experiment is what I want to test. I want to test the effects of my study guide. My study guide is what I can change from one student to the next. My study guide is the independent variable. The dependent variable is what I expect the study guide to affect. I expect the study guide to affect test scores. So test scores would be my dependent variable. In this experiment, I'm going to have some constants. There are other things that can affect test scores. Things like the ability of my students. I have to make sure the ability of my students are relatively equal across the board. The time of day I give the test. That can affect the results on the test scores. So I have to make sure I give all my tests at the same time. It has to be the same subject matter, the same test. If I'm using a mixed group, an equal number of males and females would be important. Anything that I can think of that could affect the outcome of my experiment, that could affect my test scores, has to be a constant. I also need a control. I need something to compare my results to. I cannot give all my students the study guide. It'd be nice and I'd like to, but I can't. I have to take my group and split it in half. Half my group gets the study guide, half does not. Doing that, I have a way of comparing my results. If I gave everyone the study guide, I'd have no way of knowing if test scores improved or not. But by taking half the group and not giving them the study guide, I can compare the results to each other. I can look at the two groups side by side and have proof that a change occurred. The fifth step is to make observations. Those observations can be qualitative or quantitative. Qualitative are describing things using words, like colors, red, green, blue. Tall, small, textures like rough and smooth, hot and cold. Those are qualitative observations. Quantitative observations are numbers, measurements, and counts. The difference between qualitative and quantitative is their meaning. A qualitative observation can mean different things to different people, like the word tall. I'm 5'8" tall, so someone who's 6'5" is tall to me. But if I were 4 feet tall, someone 5'8" might be tall to me. It's all relative, and we don't like that in science. We want things to be universal. So we tend to go for the quantitative observations, the things that mean the same thing to everyone. Those quantitative observations are numbers, and numbers are best, again, because they mean the same thing to everyone. 5 feet is 5 feet, no matter who you are. These numbers can be counts or measurements. We can simply count something. A desk has 4 legs. 4 is 4, it doesn't matter who you are. Our most common measurements, and the basic measurements in most sciences, are length, mass, and volume. Length is a distance between 2 points. It can be your height, it can be width, it can be length. It's measured with a meter stick, and the metric base unit that we use is the meter. Mass. Mass is the amount of matter in an object. How many atoms and molecules are there? We have nothing to compare this to in the English system. There is no mass that we use in the United States. Mass and weight are not the same. Mass is measured with a balance. Balance is measured in grams. Gram would be the metric unit. Volume, the third of our basic measurements, is a measurement of how much space matter occupies, how big or small something is. Volume can be directly measured with things like a graduated cylinder, or it can be calculated using formulas like length times width times height. For our direct measurements, the ones that we can take with things like graduated cylinders, the metric base unit is the liter, and everything else would be derived off that, like milliliters. For the calculated ones, we end up with cubed units, like cubic meters or cubic centimeters. The sixth step in the scientific method is to analyze the data. When we analyze, we're trying to understand the data. We're trying to make sense of it. During the experiment, when we're running the experiment, all we want to do is record the data. We don't want to try to analyze it as we're running the experiment. The reason for that? Bias. If we're trying to analyze the data as we collect it, we might change our numbers to reflect what we think should be happening. For example, we're measuring temperature, and we're measuring it every 2 minutes, and after 2 minutes it's gone up. After 4 minutes it's gone up. 6 minutes it's gone up. And at the 8-minute mark it goes down. We might think we did something wrong if we're thinking about that increase. All we want to do is record. We wait till the experiment's done, then we analyze. We try to figure out what the data is telling us. To do this, we might be doing some math, averaging, finding the mean, the mode. We might be making graphs. Line graphs and bar graphs are the two most common type that we make in science class. Line graphs when comparing two sets of numbers or two sets of data, looking for trends in that data. Bar graphs when we have one set of numbers and a set of discrete categories like days or months or things like that, or names. Data tables are also very useful. It helps us organize that data in a way that we can see trends. We can see what's taking place. One of the most important things to remember about analyzing data is the position of the variables on something like a line graph. Again, a line graph is used when we're comparing two sets of numbers, two sets of data, and we're trying to find a trend in that data. When we make these graphs, we'll put the independent variable on the x-axis. We'll put the dependent variable on the y-axis. We'll do that all the time. That way, if somebody's looking at our graphs, they know what we did. They know we changed this to see what happens to this. They can kind of figure out what our experiment was without ever really looking at the experiment. We do need to understand the trends in these lines. For example, this one is showing us an increase. As our independent variable increases, our dependent variable increases. The straight line is what we call a linear increase. If the line were sloping down, we call it a linear decrease. The final step in the scientific method is to draw that conclusion, figure out if your hypothesis was right or wrong. In this last step, we have analyzed the data already. We know what the numbers are telling us. We look back at our hypothesis and we simply say whether or not we were right or wrong. If we were right, pat ourselves on the back. We did a good job. We guessed correctly. If we were wrong, no big deal. If we were wrong, we go back and we choose another hypothesis and we try again. We try until we get the right answer. An incorrect hypothesis is just as valuable as a correct one. Again, all the times that I worked in the science labs and did research projects, I was wrong more times than I was right. The incorrect hypothesis is valuable. The incorrect hypothesis does something. What it does basically is eliminate one of your possibilities. You know what the answer is not, allowing you to move on to something else. That is the scientific method in a nutshell. Obviously there's more details in this, but it should give you a rough idea of what we as scientists do.